biotransformation, also known as drug metabolism. In some cases, biotransformation is referred to as detoxification, but of course, it is not all forms of biotransformation that results into detoxifying substances. In actual fact, in some cases, biotransformation rather results into toxification. That is, rather making a substance which wasn't toxic or maybe less toxic becoming even more toxic after undergoing biotransformation. So we're going to look at not only in just the biotransformation that occurs in the body, in biological systems per se, but I will also relate it to the ecotoxicology, say biotransformation in the environment. Briefly, we'll look at some of these aspects for you to appreciate some, extra aspects of this topic. Now, when you take the process of digestion, for instance, that is that you take in food and the food gets into your gastrointestinal tract and undergoes various processes of breaking down, and then nutrients get absorbed into the bloodstream. In this case, what we are referring to as biotransformation is the metabolism of, say, chemical substances that get into the body. And where these chemical substances get metabolized is mainly in the liver. So we are basically looking at the processes of metabolism of chemical substances that get into the body. Now, when you take any substance which we refer to as a xenobiotic, a substance which is coming from outside of the body, that is from the environment into the body. That substance, if we take the external part of this rectangle, the box, as the environment of the organism, and inside the rectangle is the organism. Substance coming from the environment into the organism, meaning the organism is is exposed to a certain substance. That substance, first of all, gets absorbed into the bloodstream. That is, if the substance is taken even by oral ingestion, for instance, the substance, first of all, in the digestive system gets absorbed in the small intestines into the bloodstream. Even if it is, say, coming through the skin or different routes. We'll look at the routes of entry of substances. But generally, if the substance gets into the body, first of all, the substance gets absorbed into the bloodstream. So that is absorption. And then undergoes the process of distribution. That is where the substance now goes to different organs and tissues of the body. Now, here you can have the substance going through two paths. So in that process of distribution, the substance can get bound to some plasma proteins, or as the substance is distributed to tissues, the cells of the tissues have some receptors on them. And so the substance can get bound to the receptors of these cells. Those are, say, target cells. and the binding of the substance to those receptors on the target cells will result in a therapeutic or a toxic effect. So even if the substance is a medicinal substance, depending on the dosage of the substance, it can cause either a therapeutic effect or a toxic effect. Or if the substance is a toxin, it will cause a toxic effect. Now, the other route that the substance may pass is that the substance, mainly in the liver, undergoes metabolism, which is biotransformation, and the metabolites, that is the substances that are formed from the process of biotransformation. Those metabolites now have a therapeutic or toxic effect on the body. And then further, you can have excretion of the substance and its metabolites out of the body, mainly through urine. You see there is an arrow that curves around the biotransformation, only showing that here to after distribution to various tissues and organs, the substance may just directly have a therapeutic or toxic effect on the cells, the target cells and the tissues that the substance gets to, and then gets excreted. Or even after biotransformation, the metabolites from that process of biotransformation can bind with receptors or even plasma proteins and then have a toxic effect. So these are the various pathways that a substance that the body is exposed to will go through, as a general view of what happens with xenobiotics, chemical substances coming into contact with the body. Now we look further at various pathways that these xenobiotics can go through. So we have the first route of entry of a drug is oral, and that substance going to the body through the buccal cavity or the mouth then goes into the gastrointestinal tract. Then from the gastrointestinal tract, as the substance now can get absorbed into the bloodstream through the villi of the small intestines, eventually the blood that flows to the liver carries the substance there to undergo biotransformation. And then you have the metabolites and even some part of the untransformed drug getting excreted into bile, which is stored in the gallbladder, which then can get excreted back into the gastrointestinal tract in the duodenum and then be excreted through fecal matter or stool. Some of the chemical substances get into contact with the body through inhalation. So these are gases that through the nostrils, as an individual breathes in these substances, it can be a medicinal substance, it can be some poisonous gas. Obviously, this then goes into the lungs. Then in the lungs, the small pockets that into which you have these gas getting into to have contact with the bloodstream, get absorbed into the bloodstream, the alveoli. And then you have Some after you have maybe some metabolites that are also, gaseous that can be breathed out, then come out, excreted with the exhaled air out of the body. Or you have substance, this is a drug that is introduced into the body intravenously, and that can also undergo some filtration with the blood lymph. These then can go to the kidneys, some further filtration, and then get stored in the bladder and get excreted out of urine. Or from blood lymph, this goes to extracellular fluids that, for instance, are then excreted. It's either the substance itself or metabolites excreted by exocrine glands, that are sweat glands in the skin, part of the integumentary system of the body. And then you have the excretion of this drug through sweat. Or you can have also the substance after getting into its the target cells of the organs in which the drug has been distributed to. You have these substances then going into soft tissues, like they can have some deposits of this drug in, say, the cartilages that are giving lubrication between joints, or substances can even get absorbed into the bones. You can have some of these substances also getting absorbed into adipose tissue, and that will actually be substances that are lipophilic. They are soluble in fats and oils, and so they can easily get deposited in the adipose tissue under the skin. Or you have some drugs that are administered into the body subcutaneously, cutaneously, intramuscular, or intracavitary. You have these substances also. They can go into the blood lymph and undergo the same processes. So substances can pass through different routes of entry into the body, and once they get into the body, they pass different pathways to different organs and tissues, undergo different processes of transformation, and eventually the substances themselves and their metabolites can get excreted out of the body through different means, such as fecal matter, exhaled air from the nostrils, urine, sweat, or they get accumulated, deposited in soft tissues like cartilages, and get also accumulated in bones or adipose tissue under the skin. Now, when you take any drug from the pharmacy and you look at the leaflet in these drugs, there's a portion that talks of pharmacokinetics. Actually, I think you did pharmacology, or if you haven't done it yet, you do it. Pharmacology is divided into two main parts, which is pharmacodynamics and pharmacokinetics. So any drug that you take, actually, if you look at the leaflet inside, you may find these two components of pharmacology, something written on them, which is pharmacodynamics and pharmacokinetics. And pharmacodynamics will talk about the effect of the drug on the body, like the person who takes the drug, what the effect of the drug is on the body will be tackled under pharmacodynamics. Then pharmacokinetics will talk about the processes that the drug will undergo. In other words, what the body makes of the drug. It's almost like the effect of the body on the drug, because when the drug enters the body is like that processor that will then either break down the drug or, have various processes through which the drug will go to get it transformed. And all those processes are what we refer to as pharmacokinetics. So like what we saw earlier, which was absorption, distribution, metabolism, which is biotransformation. That is actually what we are looking at. And then excretion. So you have, pharmacokinetics made up of these four components: absorption, distribution, metabolism, and excretion. So in other words, what actually we are looking at in this in this topic is just one aspect of pharmacokinetics, which is biotransformation or metabolism. We are not looking at the process of absorption of say drug substances. We are not looking at the process of distribution of drug substances, and we are also not looking at the processes of excretion of these substances. We are only looking at the process of metabolism, which is biotransformation of the drug. Now, these two representations that you see are showing the movement of a drug taken into the body. So it's either so that we are having two curves on the very first curve, which is A, and the second B. Actually, they represent the same things, except that you have on the vertical axis in graph B, having the values of concentration. They are logarithmic values, the logarithmic values of concentration here. But in graph A, it is just the concentration values themselves that will be on the vertical axis. Now, usually you use the logarithmic values of concentration on the vertical axis when you are dealing with minute concentration values that representing them directly on the graph can be difficult because you find difficulty with having consistent intervals between these concentration values, and so you convert them into logarithmic values so that you can make more meaning out of it. But here, on the A, that is directly the concentration values, and the first curve is a curve that comes from the say the highest concentration value and coming down all the way. So the graph is concentration against time. So over time when the drug is taken. So this first curve, the curve which is curve 1, is representing the pharmacokinetic of a substance that gets into the body intravenously. In other words, it gets directly into the bloodstream without going through the say if it is oral, it's going to go through the digestive system. But this curve A is representing a substance that gets into the body intravenously, directly into the bloodstream. And so because it gets directly into the bloodstream, the concentration is very high right from the beginning. But as the drug undergoes the process of distribution and biotransformation, the concentration of the drug reduces all the way until you have elimination, which is the beta phase. and you have the elimination of the drug to the end of the time of observation of the concentration of the drug in the bloodstream. Now, the second curve, which is curve 2, represents the pharmacokinetics of a substance which is taken into the body by oral administration, so that because it's oral, when the drug is taken in over time, in the gastrointestinal tract, specifically in the small intestines where the drug is going to get absorbed. Right from the beginning, the concentration of the drug is very low. And when the drug starts to get absorbed in the small intestines, there is that steep increase of the concentration of the drug all the way until it reaches maximum concentration. And because the drug will also be undergoing biotransformation, from then on you have the breakdown or the metabolism of this drug occurring, and so the concentration of the drug begins to drop over time, gradually all the way until it reaches the beta phase. That is the elimination, the elimination time of the drug. So you have the drug excreted out of the body, and until there is the blood is completely excreted out of the body, and you no longer have it in the bloodstream. So, I mean, this is to show the movement of drug substances in the body, comparing, you know, the path of the drug through oral administration and then intravenous administration. How about, you know, drugs that are taken into the body, say with repeated doses at regular times? So when you take a drug, say you should take it maybe one in the morning, one in the afternoon, one in the evening after meals. You are given some prescriptions like that, or you take the drug one in the morning, one in the evening. So you can tell that the curve that we are seeing for repeated doses of a drug is representing a drug that is taken into the body by oral administration, so that at the beginning the concentration of the drug is very low, and as it gets absorbed, the concentration increases, it goes up, and then it begins to calm down. That is when the drug starts to undergo biotransformation. Then it reaches a point that a second dose is taken. So you have these arrows at the top showing times of doses. So the first dose is taken down at this time. The second dose, that is the second D at the top. When it's taken at that time, then the concentration increases again, up. And just when it begins to come down because it's undergoing biotransformation, it reaches a point, and the second dose is taken, then the concentration increases again. So you notice this oscillation of the concentration of the drug within a certain interval, which is, you know, the concentration interval, or the window, what we refer to as the therapeutic window. So between the highest concentration level and then the lowest concentration level that the drug reaches before the next dose is taken. That interval is a therapeutic window, meaning that the concentration of the drug in the system needs to be within that interval in order for the drug to have a therapeutic effect. That is the reason why those times are given, because the developers of the drug estimate that after taking the drug over a certain interval of time, the concentration of the drug reaches a point that it has to be increased again to reach to remain in the therapeutic concentration interval. So that if the concentration goes below that therapeutic window, then the therapeutic effect of the drug is not felt. At the same time, if there is an overdosage of the drug, it goes beyond the therapeutic window, goes above it, and then it means that the concentration would have gone into a toxic level, so that it begins to have a toxic effect and no longer a therapeutic effect. So you see such a pharmacokinetic of drug substances that are taken with repeated doses, at regular intervals as well. Right. So you see the interval which is between each of the times being the same. It's a regular interval. If it's 2 hours, it's 2 hours for all. If it is 3 hours, it's 3 hours, 4 hours, like that. So that is what you see being represented on this pharmacokinetic curve. Now, okay, there's one straight line which goes through the curves. I mean, this is just to show the average dosage of the drug around which you want the concentration to oscillate for the drug to remain in its therapeutic window. Right. Now, you see, substances taken into the body can, as I was saying earlier, can either have therapeutic effect or toxic effect. So, in fact, in toxicology, it is said that toxicity of a substance depends on its concentration. So that you can have a substance which is not toxic, known as being toxic, but actually at a certain concentration, that drug that substance can be toxic. It is only that when the substance is below, a certain threshold, then that substance does not pose any toxic effect. So we see, like drug substances, common drug substances that are on the market, to say, okay, and so we look at concentrations of these drug substances, the differences in the concentrations that can have either therapeutic effect, toxic effect, or even lethal effect, that is even resulting into death. Common over-the-counter drug like paracetamol will have a therapeutic effect between concentration ranges of 10 to 20 milligrams per liter in the blood. But when it passes, say, 400 milligrams in the blood, that's you can begin to have paracetamol toxicity. And if it goes above 1,500 milligrams per liter, it can result into fatality. I mean, that similarly, drugs like aspirin, phenobarbital, chlorpromazine, diphenhydramine, nitrazepam, all of these have concentrations that are therapeutic, and some concentrations that are toxic, and some concentrations that are lethal. I mean, this is not to say these concentrations represent, for instance, what is written on the dosage form. Say, for instance, if you take paracetamol and you see maybe 200 milligrams, and you take in 2 tablets of that, you don't think that all that is going to get absorbed directly into the blood at once. No, you remember that when you take it orally, it goes into the bloodstream. All of it does not go into the bloodstream at once. It gets absorbed gradually, and even as it is getting absorbed into the bloodstream, it is getting metabolized at the same time. It's undergoing biotransformation, and excretion will start occurring, so that at any point in time you do not have the concentration of paracetamol at that concentration, which you see written on it. Say this is the amount. Say you have 400 milligrams, or say 200 milligrams, and you take 2 tablets. Not all of it is going to get into the bloodstream at the same time. In any case, the volume of the blood is not 1 liter. So if you say milligram per liter, you don't get confused with that to say, how come we say that therapeutic effect is just 10 to 20, but what you see written on the drug is say about 200, which you are taking, say, 2 tablets of, which is equivalent to 400. So is it not toxic? So you have to look at these facts, to these aspects, to get to have such an understanding that yes, the tablet you are taking, that tablet is telling you that when you take one of it, you are having, say, 200 milligrams of the active pharmaceutical ingredient in there, but it doesn't mean all of that gets absorbed into the bloodstream all at a time. But the absorption occurs, gradually, and even as the absorption is occurring to the bloodstream, you still have processes of biotransformation and excretion occurring at the same time, and so you do not have all the drug in the blood all at a time. Right. Now there can be different factors that will influence the choice of the route of entry of a drug, and some of these are, say, age, gender, body weight. So depending on the age of an individual, a certain drug, maybe an old person who has difficulty with swallowing will not be given a drug that will require that he swallows it. Even if it is so, maybe they will be given the liquid form, either it's a syrup or suspension, instead of a tablet. Gender, of course, there are some medicines for women, some medicines for men. Body weight, yes, some drug substance, some drugs are calculated, the dosage are calculated depending on body weight. Similarly, dosage, again, apart from age, what I talk about, say, swallowing, for instance, in terms of age, the dosage which is given to children is different from dosage that is given to adults. That is a common thing you see on drugs, medicines that are having these inscriptions on them. They give you the dosage which you should give to adults, and the dosage which you should be giving to children. Then you can also have the choice of, route of entry, also depending on some, chemical properties, call it physical chemical properties of these drug substances, being, say, the molecular weight, the size of the chemical compound, state of aggregation, whether it's a solid liquid, is it a colloid, is it some suspension, some spray, gas. You have in the process of drug development, have these properties considered, in terms of choosing the route by which these drugs should be taken. Depending also on the type of condition that the drug is being developed for. So you have some inhalers that will deal with, say, respiratory tract issues. You can have common tablets, which are solid. You can have liquids, which can be syrups, suspensions. You can also be having some suppositories, which will have the route of entry being anal. So you have such properties influencing the route of entry of the drug. You also have, things like the temperature or humidity of 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 the environment also influencing that. So, for instance, for in some areas where you have maybe very dry conditions, for instance, when you maybe you want to take some drug which should be thermal, for instance, where you have humidity being very low, stuff like that. So you have to keep the skin moist in a way before you can actually apply such drugs on it. Things like that. Temperature can also influence, because the storage of these drugs will also be considered in a way. Quantitative characteristics, that is, how much time should an individual that is taking the drug be exposed to it, is considered in the development of a drug, so that when a person takes a drug, how much time will it take for that drug to get excreted out of the body, and actually how much time is it that is needed for the therapeutic effect to be felt. All those things are considered, because if the exposure time is too high, it may begin to cause some adverse effects. If the exposure time is too short too, then it means that the therapeutic effect is not to its optimum. So all of these parameters are considered in drug development. Right. Then another is the route by which the drug is being taken. How much of its surface is exposed for absorption of the drug. The skin is the largest organ in terms of area. As you see, the skin is occupying 1.2 to 2 meters squared. So that's the substance that is applied on the body, or even if it is from the environment that is getting penetration through the skin into the body, you have large surface area, and that should inform the dose, how much of that substance should be put on the skin, because over such an area, it can be calculated how much of the drug can get into the bloodstream at a time. The oral cavity, how much of it, the mouth is small. I mean, that is like say 100 times smaller than the area of the whole skin. For instance, how much of a drug substance can get absorbed through the oral cavity. The stomach, small intestines. So actually, the small intestines they are, because they have these villi, the small foldings that are adapted for the highest amount of absorption. So you can see that is 100 meters squared. That's very large. So even though I said the skin as considered as the largest organ, you see that the intestine, the small intestine, is actually having the largest surface area because of the foldings that is in it. So you can see all these. The lungs, the lungs also have the alveoli, the small pockets with capillaries that have, gaseous exchange with the blood. Also, are having about 70 meters squared, which means substance which is taken in through the nasal cavity, when it gets into the lungs, gets absorbed very quickly, faster than say when it is being taken orally. All right, so we can continue and look at. So after you have the drug getting to the tissues, these drug substances will have some interaction with the cells that form the tissues. And some of these interactions, the interactions include what we refer to as membrane transport. That is the movement of the drug across, say, the cell membrane. And even some of these substances, after they get into the cell membrane, can even go across membranes of organelles, like the nucleus, endoplasmic reticulum, the Golgi apparatus, etc., etc. And these processes of membrane transport are different. You have processes of simple diffusion. I mean, that is just movement across the cell membrane, flowing with the concentration gradient of the drug. So from a higher concentration of the substance to a lower concentration of the substance flows freely through the cell membrane. Then you have what we refer to as facilitated diffusion, which you see the green boxes here are moving through the membrane protein. So it's diffusion, but it's not, the substance is not moving through the cell membrane freely. As you can see, the substances that are diffusing, that are moving by simple diffusion across the cell membrane, are depicted to be smaller. They are smaller and could easily move across. In facilitated diffusion, these substances are bigger, and they needed to go through a membrane pore, which is a protein across which that protein becomes like a transport protein through which you have the substance passing through into the cell. Then we have active transport. So in active transport, you see, even though it also involves a membrane protein, this transport protein embedded in the membrane, the difference with the facilitated diffusion is that you actually have, with active transport, the participation of ATP, or there is some energy that is dissipated in this process. So you can see ATP is involved in phosphorylation of that protein, the transport protein that will activate the transportation of the substance to occur. Usually what will happen is that when you have one phosphate group from ATP activating the protein, that is sort of opening that transport protein for that protein pore for the substance to go through it. The energy is required for that movement of the substance across the cell membrane. That is why it's being referred to as active, because of the expense of energy in that process. Well, you can also pay attention to the structure of the cell membrane itself, where you have the cell membrane being a bilipid layer, consisting of phospholipids. On the external parts, you have the hydrophilic part. I mean, if you go back and look at the structure of fatty acids, the carboxylic acid groups, those are hydrophilic, whilst you have the hydrophobic chain, that is the long carbon chains attached to this carboxylic acid carboxylic functional groups. So these are the ones that are represented green and as the hydrophobic non-polar fatty acid tail of these phospholipids. So you can pay attention to the structure of the membrane as well. I think you did something on biomembranes along the line. Those of you who read SLT, did HND SLT before coming to do medical laboratory technology, there was a course on biomembranes. So I think you would have seen this. It's possible even those of you who are doing directly, say medical laboratory technology, will also may have encountered this. Maybe in cell biology, for instance, I'm sure there will be something like this. Okay, then you also have the transport of solid substances, where you have the cell membrane actually engulfing these substances. For much larger, bigger solid substances, you have the process of phagocytosis, where you have the cell membrane engulfing the substance and then forming what is referred to as a phagosome or fold vacuole, which will result into the breakdown or dissolution of these substances. Usually these processes that we are seeing here, the phagocytosis, pinocytosis, they will be occurring much more with unicellular organisms, than say in eukaryotes. And then, so what you see between phagocytosis, pinocytosis is that the pinocytosis, again, you see that what the substances that are engulfed in the cell membrane, are much smaller, forming these vesicles that will result into the breakdown or the metabolism of the substance in these vacuoles. The third type of transport we see here is, this cell membrane having receptors on its surface, and these receptors on the surface recognize some substances, and so which means that they are going to interact with specific substances and not others. So that if you have a mixture of these, blue boxes and these orange stars representing different substances that are present, the receptors recognize only these orange stars, which are the types of substances that the receptors recognize. In other words, the target cell to which the drug is actually recognized by these receptors. Now, those receptors can be some membrane protein. It can be some membrane lipid, some complex. It can be glycolipid, lipoproteins that are interacting with the substance, and it is only, so you see, it is only those substances, the stars, that are engulfed into that vesicle for it to undergo digestion, or call it undergo transformation. Right. So that's about membrane transport. Now, when you take the process of biotransformation, it's divided into two phases. Actually, we've seen at least one of these much earlier when we were looking at hemoglobin, for instance. That's the process of conjugation. We will see it again to remind ourselves about it. So we have biotransformation divided into two phases, where It's either when you take in a substance, the substance can go directly to just the second phase. That's the phase 2. The substance may not go through a phase 1 reaction. It will just go directly to a phase 2 reaction, which is conjugation. It means that the substance gets bound onto another substance. Usually what happens is that many of these, say, drugs, medicinal substances that are taken, the active pharmaceutical ingredients are lipophilic. They are fat soluble. But, in order for these fat soluble substances to get excreted out of the body, and that excretion usually occurs through urine, and urine is aqueous. It means that the metabolite or the substance needs to be converted into a hydrophilic substance. And the conjugation, the combination of the drug with another substance in the body, usually an endogenous substance, or sometimes it can just be conjugation with a functional group. It turns the substance into a hydrophilic substance so that it can get easily excreted out of the body. So you see on these vertical lines it's been written absorption, biotransformation, and elimination. So these are the phases through which, these are the stages through which you have the substance passing through. But then you have the 2 phases of biotransformation being either you have conjugation as phase 2, or you have the drug can undergo phase 1 reaction, which will result into metabolite. So that is if the substance undergo some biotransformation reactions, you have new substances forming out of it. Now, those substances can either be active or not active, inactive, before the substance then go to the second phase, which means that metabolite now gets conjugated to another substance, making it hydrophilic and gets excreted out of the body. Now, the last one you see is the same as what you see at the top. So it's either you have the xenobiotic going directly to the second phase, which is conjugation, to get excreted, or you can have it undergoing phase 1 reaction and also phase 2 reactions. Now let's look at these phase 1, phase 2 reactions more closely. Now, phase 1 reactions are mainly three types of reactions. These are oxidation, reduction, and hydrolysis. So you can relate to it as redox reactions. They are phase 1 reactions. Redox reactions means oxidation and reduction reactions. They are phase 1 reactions plus hydrolysis. Hydrolysis is also a phase 1 reaction. So we see examples of these oxidation, reduction, and the hydrolytic reactions given here in the first column. Oxidation reactions include hydroxylation, that is a substance to which you have a hydroxyl group getting attached. Decarboxylation will be removal of a carboxyl group. Desulfurization, removal of a sulfur group in there. Formation of oxides is oxidation. Dehalogenation, removal of halogens. Oxidation of alcohols, which we will see much in detail. Oxidation of aldehydes also you see together with oxidation of alcohols. And all of these are occurring in microsomes, in other words, in endoplasmic reticulum, and also in the cytosol, that is the cytoplasm. Reduction reactions, reduction of aldehydes. You have azo reduction, and then reduction of nitro compounds. Some of these we'll look at them much more in detail when we start to look at some examples of biotransformation of some specific substances. Hydrolysis, splitting of esters. So, for instance, the breakdown of if you take even inside the body, it's not even a drug substance that you are taking in. If you break down, say, triacylglycerol, you break away fatty acids from the glycerol. It's a form of splitting of an ester, because the bond that is between the glycerol and the fatty acid is an ester bond. Fatty acids are esterified to the glycerol backbone. Breakdown of amide bonds, that's almost like metabolism of drug substances that are peptide-like. And these occur also either in the endoplasmic reticulum, in the cell, or in the cytoplasm. So if you don't remember anything at all, at least you should remember that phase 1 reactions consist of oxidation, reduction, and hydrolysis, even before you go to look at the various examples under these 3 processes. Now, an example of so we start to take a look at some specific examples of biotransformation, and this reaction of N-dealkylation, removal of an alkyl group, will be falling under phase 1 reaction. Now, what we see here is, one common painkiller, strong painkiller, codeine, which either, undergoes some dealkylation to form a metabolite, norcodeine. Now, when you take a critical look at the structure of codeine, you see there is an alkyl group. This is a methyl group on this nitrogen here. You see CH3, a methyl group there. But in norcodeine, you see in place of that CH3 is just a hydrogen. Means that alkyl group has been removed. That is dealkylation, removal of an alkyl group to form norcodeine. And even further, if you look again at the other path of the metabolism of codeine, the formation of, say, morphine. You see that again, there is an alkyl group, there's a methyl group here, a methyl group up here. But in morphine, in place of that methyl group, you have a hydroxyl group there. That is some, dealkylation, and then at the same time, there is oxidation occurring. In place of that CH3, you now have a hydroxyl group. So you can look at it as an oxidation reaction, specifically hydroxylation, as an addition of a hydroxyl group there. Then you also see that even morphine can get converted into normorphine, and that too is occurring by dealkylation of morphine, where you have the alkyl group, this CH3 on this nitrogen in the ring is removed, and so you now have just a hydrogen there on that nitrogen. So that is another dealkylation process. Now, one interesting thing about this process is that, you see, right from the beginning, I mentioned that even though biotransformation sometimes is referred to as detoxification, it is not in all cases that the process of biotransformation results in detoxification. Actually, in some cases, the process can result into the formation of more toxic substances. And here we see an example where, in fact, codeine being transformed into norcodeine. Norcodeine can be about 6 times or more, toxic than codeine and morphine. In fact, the nor metabolites, that is normorphine and norcodeine, are more toxic than their precursors, the substances from which they are formed from. What it means that norcodeine is more toxic than codeine, and normorphine is more toxic than morphine. So that is an example of biotransformation that results into metabolites that are more poisonous than the substances themselves. So it's not detoxification there. That is toxification. It's actually the reason why, earlier on, this process of biotransformation was just referred to as detoxification, because it was being related to as a process by which substances that get into the body, get neutralized. But as more studies reveal that you can actually be having toxification even in that process of transformation in the body, then the term biotransformation actually became more appropriate than detoxification. Now, another common phase 1 reaction is oxidation of alcohols and aldehydes, right? What we saw in the table, a specific example. So this is ethanol, and under the activity of alcohol dehydrogenase, the enzyme which is responsible for the metabolism, or the, say, oxidation of alcohol into their aldehydes. So you have alcohol dehydrogenase together with its coenzyme NAD+, which actually gets reduced. NAD gets reduced, and then in that process you have ethanol getting oxidized into acetaldehyde, and the reaction goes further, where acetaldehyde, so that's an aldehyde again undergoing. So aldehyde oxidation is also occurring here, where you now have the aldehyde dehydrogenase as the enzyme that is responsible for the conversion of acetaldehyde into acetate. Again, the coenzyme that is acting there is a nicotinamide dinucleotide. Now, further, I think I mentioned this in one of our earlier lectures, that acetate can then go into the synthesis of acetaldehyde, into the synthesis of acetyl-CoA. And that acetyl-CoA then can go into the Krebs cycle and generate energy. And it's one of the reasons why alcohol, even though it's having 2 carbons, and is expected to be more poisonous than methanol, which is having just 1 carbon. But alcohol is rather less toxic, because the body is able to metabolize ethanol into acetate, which then forms acetyl-CoA, and then just forms energy. But of course, excessive consumption of alcohol will mean that there's going to be an accumulation of acetaldehyde, because the rate of consumption could be higher than the rate of metabolism of the ethanol. And when that happens, you find that acetaldehyde begins to accumulate in the bloodstream, and that is what results into person's head staining, head aching, and persons, such persons that consume so much of alcohol begin to lose orientation so that they are not even able to walk straight sometimes. And even in some cases, excessive consumption, of course, if there is going to be accumulation of even acetates, which means there's going to be excessive formation of acetyl-CoA. What will then happen is that this excess amounts of acetyl-CoA will begin to form acetoacetate. These are ketone bodies. So you realize that those ketone bodies is they are also indicators of excessive energy molecules in the blood. So one of the reasons persons with diabetes are advised not to take alcohol, because alcohol will be adding to acetyl-CoA as even excess glucose when it goes through the glycolytic pathway will also be generating acetyl-CoA. So in other words, the products of glucose will be the same as the product of alcohol that are being consumed in terms of cellular energy. And so consumption of alcohol will only go in there to aggravate the diabetic condition. That's why persons with such conditions are advised strongly against the consumption of alcohol. Then another example of a phase 1 reaction is to do with the drug antipyrine. So at the top, the first molecule is labeled I. That is the drug itself, antipyrine. And then we have different metabolic pathways that this drug can pass. And here you realize the very first metabolite, which is labeled II, and the name is on the right hand side. You can see that is 3-hydroxymethyl antipyrine, and we see that there is a hydroxyl group. It's a hydroxyl group that is attached, so that is hydroxylation. That process is hydroxylation. That is one of the phase 1 reactions that you see. So it means that the very first metabolite is formed through the process of hydroxylation. I mean, if you just try to take a look at the differences in the molecules. So if you look at the antipyrine itself, there is no hydroxyl group on the CH3 at the top there. There is no hydroxyl group on it. Then when you come down to II, you see that on that CH3 there, you now have CH2OH. That is hydroxylation. If you take III, which is norantipyrine, you also see that there is the loss of CH3 on this nitrogen on the left-hand side. That is in the first ring at the top. The CH3 there is lost. So it means that is you have the third compound, which is III, which is the second metabolite coming from antipyrine, being formed by the process of dealkylation. Then you also, because there is that loss of a CH3 on N, then you also have IV, which is the third metabolite. Also, when you pay attention, you realize that there is a hydroxyl group that is attached on that top ring. Yeah, there's a hydroxyl group. So that also is hydroxylation. Of course, the name is 4-hydroxy antipyrine. Then V, again, you will notice that there's a hydroxyl group. At this time around, the hydroxyl group is on the lower ring. And so that is also formed through the process of hydroxylation. And so that is a 4'-hydroxy antipyrine. If you look at the metabolite that is labeled VI, that too, you see there is a further oxidation of the carbon that was having the CH2OH is now having a carboxylic acid group on it there, which is COOH, and that is a carboxy antipyrine. And so that is formed by oxidation, which is one of the phase 1 reactions. We also see that VII, that very last metabolite there, is formed from either IV or V, so that if it is IV, that is having a hydroxyl group on the ring at the top, there is a second hydroxyl group which is added on the ring that is below, and so you now have 2 hydroxyl groups on the molecule, and so you have 4 prime dihydroxy, 2 hydroxyl groups, dihydroxy antipyrine. If it is coming, if that dihydroxy was being formed from V, then of course the lower hydroxyl group is already there, so it is the top ring which is going to get hydrolyzed to have the second hydroxyl group. I mean, this is just for you to see that a drug that gets into the body goes through different metabolic pathways. It is not as just one straight pathway that the drug passes. Different metabolic processes, occur with a single drug, so that there are different metabolites that are formed, along the line as the drug undergoes metabolism. So that is the purpose of it, for you to appreciate that fact. Then you have phase 2 reactions. Which is, I mean, phase 2 reactions, we said it was conjugation. So whatever it is going to be a functional group or some endogenous substance that is getting combined with the substance that gets into the body to get a hydrophilic metabolite that can get excreted out of the body. So you can have examples like conjugation with glucuronic acid, which we saw when we were looking at the metabolism of hemoglobin. We will look at it again to remind ourselves that is occurring, in the liver. And you have conjugation with sulfates, acetylation, conjugation with an acetyl group, glutathione conjugation, that is conjugation, that is the molecule glutathione, which is also endogenous, that is binding to the substance, methylation, addition of a methyl group. So you have such conjugation reactions as phase 2 reactions that occur either in the microsomes, that is in the endoplasmic reticulum, and or the cytosol, the cytoplasm. Now, this is another common one here. Phase 2 reactions. We are looking at the metabolism of the common over-the-counter drug, paracetamol. And here too, you see you have different pathways that paracetamol can pass. So, but all of it, you can see clearly there are conjugation reactions where you have paracetamol getting conjugated to glucuronide. So that is a process of glucuronidation. And you see that about 50 to 70% of the drug actually goes through that pathway. You can also have sulfation. About 25 to 35% of the drug can undergo that process of sulfation to form paracetamol sulfate. Some oxidation reactions can also occur, and clearly that is not a phase 1. It's not a phase 2 reaction. That is a phase 2 reaction. So see, there can be a mixture. You realize we saw that some substances will have both phase 1 and phase 2 reactions. So this is one example of them. You see phase 2 reactions and you see phase 1 reactions as well. And then, if you have paracetamol taken in toxic amounts, for instance, you now about 5 to 15% of that substance can go into the formation of what we refer to as protein adducts. So you have these complexes that are formed with paracetamol with plasma proteins that can result into paracetamol toxicity. Further, you see again there is conjugation with glutathione. So you have that conjugated product also there. Even in the brain, you have, so for instance, deacetylation is also occurring here, where you have the acetyl group that is on the nitrogen there removed, and so you are just left with an amide group on the ring on the left hand side. And further, you can have conjugation of a fatty acid. This, I think, should be arachidonic acid. So a fatty acid also gets conjugated to that molecule of paracetamol. Actually, this compound, the AM404, is what is responsible for about all or most of the therapeutic effects, which is the analgesic effect of paracetamol. These conjugates with the fatty acid, arachidonic acid, which you see here below here. Right. So it's another common example of a common drug, which is undergoing various metabolic pathways, biotransformation. Another one where we saw in the table was acetylation. So this is much simpler. That this is an aromatic amine. Is a drug to which we see this is acetyl-CoA, and the acetyl group from acetyl-CoA gets conjugated with the drug molecule, so that you now have the acetylated metabolite of the drug. So that's quite simple. You can see that is, and then the CoA, the coenzyme A, it's left on this one. So the CoA loses the acetyl group to the drug molecule, and the drug becomes acetylated. And that's will result into some hydrophilicity of the drug. The drug becomes more hydrophilic and can get excreted more easily. In some cases, that acetylated metabolite also is active just as the drug itself. Now, this is what I was referring to earlier, that we actually saw it much earlier in the process of breakdown of hemoglobin in blood, where you have bilirubin binding to albumin in blood, and in the liver, you have bilirubin undergoing conjugation with glucuronide, and that process is glucuronidation. And you see you see this, what is being depicted to be happening here in the liver, that glucuronide complex with bilirubin excreted into the bile, excreted into the gallbladder, stored there in the gallbladder, and excreted from the gallbladder through the bile duct into the duodenum to be excreted out of the body. You see it, much more clearer here. the process of the conjugation, where you have glucuronic acid getting conjugated to bilirubin to give you bilirubin glucuronide, which then can get excreted through the kidneys into urine as urobilin, or getting excreted as thecobilin in stool. So you see these two pathways of bilirubin, getting excreted out of the body. But before then, you have the conjugation with glucuronic acid. We have seen this one much earlier when we were dealing with hemoglobin, when we were looking at the degradation of hemoglobin. Now, this is not biotransformation in biological system. So I said earlier that we'll look at aspects of biotransformation also in an environmental context, and this is one of them. Now you see the substance 1, which is trichlorophenoacetic acid, is a common herbicide that can undergo process of hydrolysis and forms a trichlorophenol and acetic acid. But 2 molecules of trichlorophenol can get conjugated. So what you are seeing here, hydrolysis is a phase 1 reaction, and conjugation is a phase 2 reaction. So here's an example again. You are seeing that herbicide is undergo. So this is occurring in the environment, that you have hydrolysis and conjugation occurring at the same time. So the metabolite from the first reaction, hydrolysis, now get conjugated to give you, a more toxic substance, which is tetrachloro dibenzodioxin, TCDD for short. TCDD for short. And this substance can be poisonous to some animals when they consume the grasses or the plants onto which this herbicide are being applied, or even when it gets into water bodies, it can be toxic to aquatic life. Then again, on the environment, you have another. This is an insecticide, parathion, that you can undergo process of photooxidation to form paraozone, which can also be toxic to mammals. So this is an example of toxification reaction. This is not a detoxification, because paraozone becomes more toxic than the original compound itself, parathion. And then you have also another pathway, which is hydrolysis, from which clearly you see that the sulfophosphor group over here is removed, and then you just have a hydroxy group replacing that to give you a paranitrophenol. So here too, you see 2 things happening. Photooxidation will be occurring just by exposure of the substance to, say, sunlight, and you have a more toxic substance being formed out of it. Or you have hydrolysis, where the substance is broken into two compounds. Now here also you have, so you can have the formation of say some hormones. So these are synthetic hormones, androgens, testosterone, that are formed from plant substances. This is a plant fat. Phytosterol is coming from plants. Actually, you have a lot of phytosterol in nuts. And this reaction was actually discovered with some real life occurrence of some fishes that were noticed to undergo masculinization, so that the females of that fishes begin to behave like the males. of those fishes. And it was then discovered that actually it was some phytosterol that was being discarded into the water body by a factory which was, beside, along the water body somewhere. That factory was involved in the um, manufacturing or processing of some nuts. And when they do the processing of the nuts, they release the waste into the water. And there was a lot of phytosterols in the releases from that factory into the water. But there were microorganisms in the water that could convert those phytosterols into male sex hormones, testosterone, so that you can see here that in place of a hydroxyl group that is on the very first ring, you now have the cyclic ketone group. That is some form of oxidation reaction occurring. Then you also see that in place of this side chain on the very last ring at the top, you just have a hydroxyl group. So in a way, that is just a removal, a substitution of that whole side chain with a hydroxyl group. And then you now have an androgen, which is testosterone. And that was having that effect on the fishes in that river. That's occurring, in the environment. It's biotransformation, but it's occurring in the environment, not in living systems. All right. You can have, so another example is another drug, which is cimetidine, which is used to treat peptic ulcers, heartburn, and that can also undergo different pathways. The drug is supposed to reduce gastric acidity, but then you can have some bacterial and fungal proliferation of the drug. And in combination with nitrites from food and drinks, you have the conversion of these nitrites and foods, nitrites in foods and drinks into nitrites. And you have enzymatic reactions of bacteria in the digestive system, causing the formation of nitrosamines from the nitrites from the food. So it's a drug that is being taken all right, but a drug can go and act with nitrites from some food substances, undergoing process of nitrization, and then will be forming some nitroso compounds, which will rather be increasing the risk of gastric cancer. So this can also occur. So that is another form of biotransformation, which is not resulting into detoxification. It is rather resulting into toxification. Now, just to look at some health implications and how some of this knowledge can be applied in terms of personalized medicine, knowing that the metabolic processes in organisms will not be the same. There are certain cases where you have some genetic variations, whereby maybe a drug can undergo a slow metabolism in some individuals, whilst in some you have some intermediary rate of metabolism, whilst in some you have a very fast metabolic rate in them. In these cases, these drugs, the dose that should be given to individuals should be individualized or should be customized. Other tests need to be done to know the specific dosage that should be given to an individual. If not, if the person is having very fast metabolism, what will happen is that if you are giving the same dosage that you are giving to someone who is having a slower rate of metabolism, the drug will then not have the therapeutic effect that it should have, because the person will quickly metabolize the drugs and the drug will just be excreted out of the body. Whilst if you give the same dosage that you give to someone who is having fast metabolism to another person who is having slow metabolism, the drug will then be getting accumulated in such persons' bodies and in the kidneys, in the liver, and will rather be resulting into toxicity, be giving some toxic effects if you are having accumulation into these organs. So these are, some of the considerations that need to be made with some drugs. So this is an example of a drug, say isoniazid, which is an antibiotic used for treating tuberculosis. That you see, there are some individuals that metabolize these drugs at a slow rate, some at an intermediate rate, and some at a fast rate. And that we refer to as a trimodal distribution of concentration of the drug in the bloodstream of persons to which these medicines are administered. So, generally, this is what we have done. Phase 1 biotransformation, which is oxidation, reduction, hydrolysis. As I said, if you don't remember anything at all, you should know that phase 1 biotransformation comprise oxidation, reduction, hydrolysis in the first place. Before you go to look at the examples of, some of these oxidation reactions, reduction reactions, and then hydrolytic reactions. The phase 2 reaction is just one, which is conjugation, and you have different kinds of these conjugation reaction examples of them, which we have seen, including acetylation, sulfation, glucuronidation, conjugation with glutathione. Then we also saw the fact that biotransformation has a role of not only detoxification, but actually it can also result into toxification. And of course, in some cases, a metabolite will rather be the substance which is active. In such case, it means that the substance is a prodrug that when it's administered to a person, that prodrug, when it undergoes metabolism, then is activated and becomes the substance which is having the therapeutic effect. We also saw biotransformation in environmental context, looking at metabolism of pesticide, herbicides, which can result into some ecopollutants. And then briefly, we looked at the biotransformation, health implications of biotransformation, looking at personalized medicine, looking at the importance of individualizing or customizing drug dosages for different individuals. So that is what we have gone through so far. Now the floor is open. If you have any contributions, any questions you want to ask, you can put up your hand. I will allow you. I will enable your mic, and then you can speak. So the floor is open. Set a free year.